Pharmacological control of hyperlipidemias. Well, hyperlipidemia is too much lipid content in the blood, and what's the consequences of that? Well, the consequences is atherosclerosis, which means build-up of lipids in the arteries, causing an atheroma. So let's see how that happens. Well, if you've got hyperlipidemia, then low-density lipoproteins, such as low-density lipoproteins, they accumulate behind the endothelium in the arteries, which is atherosclerosis. And what happens is that they're then oxidized by macrophages behind the endothelium. And the result of that is they're then taken up by macrophages to form foam cells. So foam cells are basically macrophages which have oxidized and taken up low-density lipoproteins. And white blood cells accumulate. And this is essentially the immune response. Now this immune response, what it does is it creates a fatty streak, and a fatty streak is the white blood cells and fatty foam cells that have been created. And then the macrophages release growth factor because as part of this immune response they release growth factor, and that causes the growth of the endothelium, and it forms an atheroma, which is a plaque over the lipid which is behind the endothelium. So an atheroma develops over it in the lumen of the arteries. And this atheroma plaque, it can rupture, and if it ruptures, you can get a thrombosis. So a thrombosis is the plaque travelling and blocking and clogging an artery somewhere, potentially causing stroke. So we can see that here, hyperlipidemia causes atherosclerosis and atheromas by immune response, creating fatty streaks and plaques which can rupture, potentially causing strokes. So as I said in the previous lecture, the long-chain fatty acids in the enterocytes, they're bound to apoproteins and packaged as chylomicrons. So the lipids, they're bound to apoproteins, they're carried as lipoproteins, so lipids and proteins, and they have a non-polar core, which is made of cholesterol esters and triglycerides, and they have a polar coating, and the polar coating is the apoprotein. And the apoprotein is required for receptor binding. You'll also see that the lipoproteins, there's a few different types of them. So you've got chylomicrons, which are formed in the enterocytes, they're the largest. You've got high-density lipoproteins, which are made of cholesterol esters, low-density lipoproteins, which are also cholesterol esters, and very low-density lipoproteins, which are formed in the liver. And these have triglycerides as well as cholesterol esters. And the transport in the body of lipoproteins, it can be exogenous, so that's the absorption, the emulsification, absorption of fats from the gut, the food we eat, into our body, and endogenous, so this is travel within the body, which can be hepatic, so in the liver, or extrahepatic, going from the liver to the various organs and tissues. First of all, looking at exogenous lipid transport. So the lipid, the fat that we eat, it's emulsified by bile acids and bile salts. It's then absorbed, it's then hydrolyzed, absorbed, and the long-chain fatty acids are packaged into chylomicrons, which are lipoproteins. And then what happens is that these lipoproteins from the enterocytes, they go to the organs and the tissues. And on the tissues, on the endothelial surface, is a membrane protein called lipoprotein lipase. It's an enzyme. What it does is it hydrolyzes the triglycerides into fatty acids and glycerol, leaving remnant chylomicrons. Now, do you remember I said that the apoprotein is the polar part of the chylomicron that sticks out on the outer surface? So what that does is it ensures the binding to lipoprotein lipase to allow the triglycerides to be hydrolyzed into fatty acids and glycerol and leave the remnant chylomicrons. And as I said, the fatty acids and glycerol, they go to muscle and adipose tissue, and the remnant chylomicrons, they go to the hepatocytes in the liver. And in terms of endogenous transport, which is hepatic, the cholesterol from the hepatocyte, it can come from two places. So remember, the remnant chylomicrons formed from the hydrolysis of the chylomicrons by lipoprotein lipase, so they go to the liver, transporting the cholesterol to the hepatocyte. And also, hepatocyte acetyl-CoA. So acetyl-CoA can come from glucose metabolism or fatty acid metabolism through the beta oxidation cycle. Acetyl-CoA is turned into cholesterol in the hepatocytes by an enzyme called HMG-CoA reductase. And this is very, very important, it's highlighted. So hepatic acetyl-CoA is converted to an intermediate MVA and then cholesterol by HMG-CoA reductase. So this cholesterol in the hepatocyte, what happens to it? Well, it's added to triglycerides, which forms VLDL, very low density lipoproteins. Remember, we said that the remnant chylomicrons go to the hepatocyte, and the cholesterol and triglycerides are packaged together to form very low density lipoproteins containing triglycerides as well as cholesterol esters. And the VLDL, they're secreted from the hepatocyte, and they again go to the endothelial surface, just like the chylomicrons from the enterocytes. The VLDLs go to the endothelial surface of various tissues, where they again meet lipoprotein lipase. And lipoprotein lipase, again, it hydrolyzes the triglycerides, and what that forms is low-density lipoproteins. And what low-density lipoprotein does is it contains cholesterol esters only, as opposed to triglycerides and cholesterol esters. And what does that used for? Well, it's used for the transport of cholesterol. Where does cholesterol go? Well, the cholesterol is transported to various cells. It's used for making steroid hormones. And it's used for forming bile acids. If you can remember, cholesterol goes to hepatocytes, where it's turned into bile acids on the smooth endoplasmic reticulum of the hepatocytes, and then conjugated with amino acids to form bile salts for fat absorption. So we can summarize that here. So the endogenous cholesterol transport, endogenous lipid transport, which is hepatic, remember it can be hepatic or extrahepatic. The cholesterol in the liver comes from remnant chylomicrons, as well as production in the liver itself via HMG-CoA reductase. And then the VDLs, which are made from cholesterol and triglycerides, they're secreted and then become LDLs by lipoprotein lipase. A couple more things to add here. Well, first of all, HMG-CoA reductase, the enzyme that converts hepatic acetyl-CoA to cholesterol, it could be inhibited by statins. So statins can be used to inhibit this enzyme, prevent the formation of cholesterol in the hepatocytes, and therefore lower the level of lipids in the blood. And secondly, lipoprotein lipase, which hydrolyzes the triglycerides in VLDLs to form LDLs, it's stimulated by fibrates. So therefore, fibrates will reduce the level of VLDLs in the blood because they'll be stimulating the lipoprotein lipase to hydrolyze them and break them down. Finally, in terms of endogenous lipoprotein transport, which is extrahepatic, so this is outside the liver. So what happens is that normal cell turnover, releases cholesterol, and the cholesterol is packaged as cholesterol esters and transported in high-density lipoproteins, which are cholesterol esters, just like the low-density lipoproteins. But what happens is that this causes the cholesterol esters to be transferred to LDLs, low-density lipoprotein, and then causes it to be taken up by hepatocytes. So here we can see that essentially high-density lipoproteins are good in the sense that they stimulate the uptake of low-density lipoproteins from the blood into the hepatocytes, which you can see here. So the HDLs promote LDL uptake by hepatocytes. And again, just to reiterate that increased HDL will increase LDL removal.